Saccharibacteria are obligate parasites that require co-culture with appropriate host bacteria. As human oral Saccharibacteria can be easily isolated from most individuals using known host bacterial species, this protocol will allow any investigator or laboratory to isolate their own strains in binary co-culture. This technique uses simple devices and equipment present in most microbiological laboratories and has been successfully used to culture over 30 isolates representing 6 species of Saccharibacteria. This protocol may be useful for the isolation of other candidate phyla radiation species, as well as Saccharibacteria from different environments and other mammals if appropriate actinobacterial hosts can be identified and isolated. Demonstrating the procedure will be Andrew Collins, a former postdoctoral fellow in my laboratory.
To begin, start cultures of host bacteria such as Arachnia propionica with enough time for them to grow to early stationary phase. To initiate the culture, inoculate 2 to 5 milliliters of tryptic soy broth containing 0.1% yeast extract with Arachnia propionica and incubate it at 37 degrees Celsius for 24 hours. Next, assemble filter holders with track-etched 0.2 micrometer filter membranes, wrap them in foil, and sterilize by autoclaving, along with centrifuge tubes and cap assemblies. Sterilize extra supplies in case of accidental contact with non-sterile surfaces. Collect dental plaque by scraping